sort of a labor of love to begin putting all of this information together, and this is in some ways our coming out party with respect to the kind of science that we're beginning to assemble on what I think is a really important topic. And when I was 17 years old, I was a freshman in college, and I was introduced to shiatsu. And it was through actually a Tai Chi class that I was taking on campus, and it was after one of the sessions that the instructor said, well, how do you feel? And I said, fine, but I have some neck issues, whatever. And she said, lay on your stomach and I'm going to do some massage. And I said, okay. So I thought she was going to start rubbing and doing what I thought was massage. And instead, she used her elbow and she began stretching me and pressing points. And I said, well, what is this? And she said, it's shiatsu. I said, okay, well, what's that? And she said, it's Japanese. Shi is finger and atsu means pressure. It's finger pressure, acupressure. It's Japanese. And it was like being struck with a bolt of lightning. I knew that this was something that I really wanted to learn. So I found a shiatsu clinic about 20 minutes from my college, and I enrolled. And I was lucky enough where, while it was run by an American couple, there were many individuals from Japan and China that were coming through the East Coast at the time. I was living in Philadelphia, who were teaching traditional Asian healing arts. And so I had a very authentic Eastern-style education. I wasn't even allowed to touch a person until I demonstrated that I could be calm and present and centered and mindful and compassionate, that there was a development process that went along with my training. And my instructors would sort of test me at times to make sure that I could listen with my hands and be receptive to all of the information that was coming out of the individual. And you realize over time that that was something very practical, that 3,000 years ago they didn't have CT scanners and EKG machines and blood labs and sort of an objective process to sort of understand what's going on with their patient. So the more attuned you are, the more that you develop your sensory skills at every level, the better practitioner you can be, which I thought was really profound. Now, of course, that was life-changing for me. I was 17 years old and in college, where it was a very difficult school, intellectually challenging, and of course I was also playing rugby on the weekends and drinking lots of beer. And that seemed to be antithetical to sort of, going on this other journey. But it really shaped how I see the world. And what you realize in some of these older healing therapies is their focus on what I would consider pattern analysis, this notion that you can sort of group signs and symptoms together and understand sort of where is the individual at today and where might they be going, even outside of objective tests and markers. So you get very good at sort of tuning in to the individual. And so pattern recognition becomes part and parcel to the intellectual sort of landscape of that kind of provider. And I know many of you in this room, probably all of you, have developed those skills as well in sort of whatever vein you've been trained in. So I then was asked to do some work in poor communities in Philadelphia and then DC. There was a lot of academic outreach in the mid-90s going into underserved populations that didn't have access to primary care. And so some of my early clients as a young shiatsu practitioner were nurses and physicians from the UPenn system, and they became very interested in my knowledge base. So I was going into churches and barbershops, teaching mindfulness, teaching touch, teaching movement and breath and nutrition in environments in which they had already been written off. These are populations that were considered sort of helpless and hopeless. They didn't have access to good nutrition. They didn't have access to safe environments. And so we brought in a whole set of resources to help serve the needs of these communities. So I was teaching these things in multiple environments and encouraging people to perform good self-care. We brought in local farmers from outside the community to sell healthy food at cost. We even went into the school system and taught teachers yoga and breath work and movement and how to run mind-body skills groups. And what we found is that people transformed. That when you begin fertilizing them and supporting them and nourishing them, that they get better. So this was, again, another level of transformation for me, that I was fully bought into the notion of sort of that holistic approach, not just in the individual level, but even at the community level, matters, and it works. So I went off and got a master's in health management at University of Michigan, and then was asked to run a large research center at University of Michigan funded by NIH. Our mandate in the 90s was to investigate alternative therapies for cardiovascular diseases. So I migrated from public health into how to run high-level academic research studies at a major institution. And then med school and residency, I sort of went to the dark side and realized that I wanted to sort of fully complete my skill set. So I wanted the MD. And along the way, I picked up acupuncture and osteopathic manual therapy and homeopathy and botanicals and so on and so forth. And eventually you sort of emerge to the other side and you realize, okay, I'm ready to be released into the wild and to take on, patient care. I was a family physician by training. And so I think in terms of these languages and models of how we understand what we're looking at. So the first doorway I walked through was sort of the traditional Chinese medicine manual therapy door. And I could understand patients through that lens. But I realized that there was a limit. And there was only so much I could do. Then I walked through the conventional medical door. And that allowed me to sort of expand what I could understand and what I could treat. And sometimes it was the right thing to do, and sometimes I felt the pharmaceuticals were the wrong thing to do. But it allowed me to see my patients through a certain lens. And then eventually sort of the functional medicine level, right? So then you're beginning to look at the mechanics of the body, and you start hearing terms like leaky gut and adrenal fatigue, and you might want to check neurotransmitters or hormone balance, that you want to tinker with maybe some level of how the body is functioning. And you realize you can, with each of these steps, you're helping more and more people. But when I moved to Virginia and I took over a medical practice there, there was still this irreducible nugget of patients that I couldn't help. They were presenting with chronic fatigue and fibromyalgia and mood disorders and cognitive decline and joint pain and headaches. And sometimes a patient had all of those, and sometimes they had some of them. But I knew that no matter what botanical, what needle, what pharmaceutical I threw at them, they just wouldn't get better. That I was looking at something that I didn't understand. I didn't have a language. I didn't have a framework. Even though with all these years of sort of learning these different ways of evaluating individuals, why couldn't I better figure these patients out? And I think we've probably all reached that limit to one degree or another. We know when that patient is showing up and we see them on the list for the day, we go, oh gosh, they're on 30 supplements and they're taking 20 medications and they've seen every practitioner and they're hoping somehow you're going to eventually help them, but you feel like they've already done everything anyway. What else can I contribute to their care process? And that can be a little demoralizing. It can be a little frustrating. And so being in Virginia, I realized that, well, maybe some of these patients, maybe some of them have Lyme disease. And you have to understand, Virginia has some of the highest rates of Lyme in the country, and the county where I practice has the highest rates of Lyme in Virginia. So we are literally ground zero. So I have more experience with Lyme disease than just about any practitioner I know, because this is all we do all day long. And I can tell you that if you're taking on Lyme disease, it is absolutely integrative medicine 2.0. You better be good at everything if you want to treat these patients. And they're just like everyone else that I had mentioned in terms of they've already tried all the supplements and they've tried the herbs and they've tried 3 years of antibiotics and they're still not better, and oh, if we just sort of kill the bugs finally, You know, they're hiding somewhere. They're in the biofilm, or they're in the L form or the cyst form or whatever it is, you know. And I thought, well, maybe they have Lyme. So I got connected with George Mason University. They at the time were developing a better Lyme test because, as you might know, our current testing for Lyme is terrible. And sure enough, with a better test, we began identifying patients who actually truly had Lyme disease without doubt. And I started treating them, but they didn't get better. Some got better, but many didn't. And sometimes I had patients that look just like Lyme disease. They smell just like Lyme disease. They act just like Lyme disease. But this better Lyme test was negative. So was it the failure of the test? Or was something else going on that was mimicking Lyme? And you think, well, what could possibly mimic Lyme? You know, if you want to send practitioners heading for the hills, you start talking about Lyme disease, because most of us don't really want to deal with it because we know how difficult these patients are. And now you're saying something else could be causing Lyme, or Lyme-like symptoms. And so being an academic as well, the program director of integrative medicine at GW, began tapping my network and trying to figure out what was going on with these patients. So we started collecting data. And our group, and some members of my group predate me in this regard, and we've been collecting data on this population for years now. More recently, that process has accelerated. We understand more about Lyme than we ever did. We also understand sometimes when you think you're looking at Lyme, it ain't Lyme. And so my job over today and tomorrow is to help you shift your frame and teach you a new language. And I know that this is likely going to be novel for you, because it's still to some degree novel for me. And we just published a study as a kind of synthesis of all of this information. So it's in the literature. I'm sure we can get you some copies if you're interested of our most recent academic article. But what I want to do is help you speak a new language for these patients that are frustrating and they're common in primary care, in your practices. They're common complaints in general. So how do we better understand what we're looking at? And so this is a model that I want to share with you that's been well validated in the literature, but it blends really nicely with sort of an integrative approach to care. And I hope that for some of you, as you begin to really appreciate the depth and complexity of this, that as you're learning to speak this new language, you can go back to your practices, start ordering some of these tests and say, oh my gosh, here it is. And I warn you, when you walk through this door, you won't go back. You can't go back. Because unfortunately, two things. One, it's common. 20% of the population. 20%. 40 million people. And number two, because of the ubiquitousness in the general population, the labs are going to start coming back abnormal. And you're going to start second guessing yourself saying, is it possible that patient after patient actually fits into this category? And the answer is absolutely. That the data is real. And some of it is because of self-selection. Who sees patients with headaches? Right? Who sees patients with fatigue? Who sees patients with digestive complaints? Who sees patients with obesity? Who sees patients with cardiovascular problems? Some of you. Who sees patients with joint pain? Sleep disturbance? I could go on and on. We are going to wind our way through physiology, and I'm going to show you how this entity, this chronic inflammatory response syndrome, often underlies and is driving this symptom complex. So that these doorways that you're coming into, where you've been told on a certain level, well, maybe it's somatic dysfunction at C1. Or no, it's leaky gut. Absolutely, I just went to a lecture on leaky gut. It's all in the gut. If you just fix the gut, they'll get better. Well, no, Dr. Heyman, I'm sorry, but I just heard this great podcast on adrenal fatigue. So it's got to be adrenal fatigue. And yeah, sure, their cortisol levels are low. That's what it is. Patient comes back at 3 months, and they're like, yep, no, I took my vitamin C, my B vitamins, and my adaptogens, and I meditated every day, and I sprinkled pixie dust, and I still feel terrible. What else you got? And you go, I don't know. And so this is how it goes, right? Like you're kind of slipping through these ideas, trying to find something that helps these patients, and yet these are common symptoms. So why aren't they getting better? This model, chronic inflammation, that underlies this enormous population is, I think, not only important, but in some ways a real breakthrough, because it begins to focus in on an aspect of our immune system that we've essentially ignored. And so some of understanding where you're at with this entity is understanding the basics of the immune system. So I'm going to go over that as well. This chronic inflammatory response we're going to cover in detail. It is not something I can give you in an hour. So today is just an overview. I'm going to start giving you some of the language for this. And we're going to start covering things that sound pretty advanced. I'm going to use terms like proteomics and transcriptomics and brain-related injury and epigenetics. So this represents a confluence of very advanced science with sort of high art of integrative medicine. And you'll see over time as we go through the lectures how we begin to sort of build your foundational knowledge, because I really want to get you to a level of confidence that you're willing to maybe dip your toe in this water. You might not feel fully ready to sort of treat these patients, but I want you to understand the roadmap and at least evaluate them to the point where you say, yep, it's a CIRS patient. They've had some sort of exposure. This is why you're sick. I see it in your evaluation process, and now let's package you up and let's maybe find you someone who can treat you, but at least I now know what's going on, okay? So even if we can just get you to that point, that would be great. What is this? First of all, it's inflammation that lasts at least 6 months, okay? It's chronic. These patients are sick and they remain sick. The second is we're invoking many different aspects of the immune system. But the most important component is going back to understanding there are really two arms of how our immune system is organized. The first is the innate, and the second is the adaptive. The innate is the early response of the immune system. It is non-specific. Okay? It's sort of ancient in its position within how our immune system responds. It's not very bright, but it's incredibly important. It's where the early troops rush in to deal with a threat. And this includes neutrophils and macrophages and microglial cells in the brain and astrocytes and all sorts of other components of the immune system that are our frontline defense. And part of that process of creating a coordinated response to an immune threat is ultimately and properly the handoff from this innate side of the immune system to the adaptive side. The adaptive side is the more sophisticated. It's the smarter side. It includes things like T cells and B cells. And T cells and B cells are very important for having that a precise response to a threat. T cells receive information from the innate side, and they do that by antigen-presenting cells. These are specialized immune cells that encounter the threat, and they take a small molecular SNP called an epitope, and they absorb it and then present it to a T cell. And the T cell is waiting to be triggered by an antigen-presenting cell. Once the T cell is turned on, natural killer cell activity upregulates, and then the T cell begins to trigger B cells. And B cells make antibodies. And these antibodies create a more specific response to that immune threat, and they create memory cells. So why am I going through all this? innate immune response, early inflammation, neutrophil, macrophages upregulated, antigen-presenting cells, so important as the bridge between the two. Well, it turns out that in this chronic inflammatory response, the people who suffer and are sick from it, they have poor antigen presentation. They don't transfer that information from innate to adaptive. They're not efficient at creating an antibody response. They remain stuck in this early innate immune activation, and therefore they remain sick for months and years. The reason why this happens is because of the person's genetics, that in fact they're born this way. And we know this because we've identified the gene sequences on chromosome 6, which house something called HLA. And among all of the other sort of responsibilities of HLA is to help the body and antigen-presenting cells be efficient at that transfer of presenting the epitope. There's about 9 genes that exist on the HLA chromosome 6 that if present, they confer poor antigen presentation. 9 genes, 20% of the population. So, whenever there's an immune threat and the innate immune system is turned on, and if it's in a person who has poor antigen presentation, they can turn on inflammation, but then they can't turn it off. That the body continually makes inflammagens and proteins from the tissue that begin to circulate through the vascular system, and ultimately they're grabbed on by the liver, they're absorbed into the biliary tree, emulsified in the bile, dumped into the digestive tract, and there they sit. And of course there's only 2 routes to go out. The first route is out in the stool as a drain. The second route is to be reabsorbed. This is called enterohepatic circulation. And so what's happening is that patients who turn on this inflammatory sequence, not only can they not turn it off, but they just keep recycling it. They keep recycling it and reabsorbing it. And so they remain in this perpetually inflamed state, and lots of bad things happen when that occurs. So, chronic inflammatory response, technically 22% of the population which carry these HLA alleles, 40 million people. And what turns this on? Here's a new term for you, a biotoxin. Usually an organism or a fragment of a living organism that has triggered the innate immune response. Certainly, tick-borne diseases can do it. So if you're infected by a tick and you've had transference of a pathogen or multiple pathogens, we know they're very efficient at turning on the innate immune system. But there are all sorts of other exposures out there that do this too. And I want to be clear, for those of you that are interested in detox, heavy metals and chemicals and organic pollutants, that is not what I'm talking about. Okay, this is not that. These are biotoxins that are triggering an immune response. Now, people who are already toxic and their cell machinery is corrupted because of the presence of mercury and cadmium and all the rest, or they've been around pesticides and herbicides and they've downregulated their enzyme substrates and their pH is low and they can't detox, sure, that's going to make these people worse. But that's not their primary issue. You will not get them better by quote-unquote detoxifying them. Okay? That's clinical pearl number 1. I see this mistake all the time. So a biotoxin is used very specifically as a term different than toxin. Okay? This is an organism that has triggered the immune response. 40 million people are susceptible. Tick-borne diseases, we know that the 2-tiered serology, meaning when you order a Lyme test, you're looking for first the ELISA and then the Western blot. When those tests were developed, they really were used more for scanning of populations as opposed to diagnosis, but unfortunately they've been picked up that way. So a recent paper demonstrates that two-tier test, the ELISA and Western blot, can generate 500 to almost 600 times more false negative results than testing for HIV. That in fact, it's only positive in a known Lyme patient 30 to 50% of the time. That you can miss up to 70% of patients with Lyme disease if you do the standard blood tests. So this is a travesty in and of itself, but it's made even more complicated when you have an individual that already has the genetic predisposition for chronic inflammation if they're poor at their antigen presentation. So there are a number of diagnostic challenges for this. Number one, does this patient meet the criteria for CIRS? I'm going to go over all of that in detail. Number two, does this patient in particular suffer from a tick-borne illness? Even harder, because our lab diagnostics for Lyme are less reliable. And then finally, what else could this be? Well, the most common other biotoxin that individuals encounter is mold. So water-damaged buildings. So they're breathing in mycotoxins, they're breathing in mold particles, and they're triggering their innate immune response. Even worse is when you get the double whammy. Patient was bit by a tick, they turned on their chronic inflammatory response, someone had made the diagnosis of Lyme, they got treated, maybe things settled down a little bit, but then now the genes are active and on, and they're, as a consequence, sensitive to the mold in their environment that they weren't to beforehand. So these things can start to stack. So, and if it's not mold or Lyme, what was or is the original exposure? And part of the diagnostic challenge is to say, is the exposure still current? Because if the truth is symptoms are generated by this chronic inflammation, we can't go on symptoms alone to know if they're still infected or they're still being exposed to mold or they have some other exposure. So you always have to ask yourself the question clinically, is the exposure today or is it historical? Because in fact, they may have already been successfully treated for their Lyme, but they're in the 20% and their body is just happily making inflammation and they can't clear it. And so the disease is actually the chronic inflammation itself. It's not the original exposure. So first case definition, 2 tiers. Is there a potential for exposure? Does this patient present with multi-system, multi-symptom illness? So headaches, brain fog, cognitive decline, mood instability, neuropathies, balance issues, digestive complaints, dysautonomias, hormonal imbalances, cardiometabolic issues. You know, it has to be across categories to fall into a CIRS diagnosis. It's not just one symptom. And that there are no confounders, meaning it's not from something else that you can figure out. That in fact they've already been through all these other evaluations and you say, well, you know what? We've done everything for you. You've seen your primary care, you've seen your specialist, you've seen your functional, you've seen your guru, you've done all these evaluations. Everybody has seen it, right? I know you have patients that come in with a notebook, right? And they've been to 20 different practitioners and they've done all sorts of tests and they're still sick. Well, you just got to make sure you rule out all this other stuff. And usually other practitioners have done that job for you. So 3 of 6 needed. And this is part of our diagnostic criteria. The first is, do they have presence of the HLA? You can measure these. I order them through regular blood work. It's HLA, DR, DQ. And we begin to sequence their genetics to say, are you in the 20%? So we want to know, do they have susceptibility? Are they poor antigen presenters? The next is a nomenclature soup of additional markers that reflect disorder in the innate immune system. Some are direct and some are indirect. One of the most important, which I'll cover in more detail tomorrow, is something called melanocyte-stimulating hormone. It's produced in the anterior hypothalamus. It's considered a regulatory neuropeptide, and it controls many different functions in the body. What happens is, as the brain becomes more inflamed as a result of this illness, and make no mistake, there's a cytokine storm in the brain. That's where a lot of this is initiated. So it's a brain on fire. And one of the consequences is first functional problems and then structural issues in the brain. So the anterior hypothalamus takes a hit, and we see decline of the brain's ability to make melanocyte-stimulating hormone. And as MSH drops, all sorts of things go haywire. And another clinical pearl, MSH, believe it or not, controls tight junctions in the gut. Oh, so here's a brain-gut connection, all you brain-gut gurus, right? Well, it turns out, you say, oh gosh, they have leaky gut, I did your food panel, and you're sensitive to 1,000 things, and I've given you glutamine and arabinogalactans and prebiotics and probiotics, and I've grown food for you, and all this sort of stuff, and you're still not better. Why is this happening? Well, it turns out that the lower the MSH level, the leakier the gut. And if you don't fix this first, you're never going to get ahead here. Okay. All right, so we're making some progress. C4a, the alternate complement cascade. This is a split product of C3 and C4. So part of the immune response in the early inflammatory process is activation of the complement system. But it turns out if you measure C3, C4, they're normal. You got to measure the alternates, because that's where we see expression of enormous inflammation. Unfortunately, right now, there's only one lab in the country that draws the C4a. And there's a sister product called C3a. And again, I'll go over it in more detail tomorrow. MMP9, matrix metalloproteinase, expressed in the vascular system, very reflective Th1 imbalances. This will start to climb in this patient population. So C4a goes up, MMP9 goes up, MSH starts to drop. And as a result, too, some of our central endocrine mechanisms that originate in the brain also start to alter. So patients begin to have a decline in antidiuretic hormone, which means that they're urinating frequently because the kidneys are not getting a signal to resorb water appropriately, so they become hyperconcentrated. So therefore, they have orthostatic issues. They'll often have electric-like shocks as they walk across carpets. And they have urinary frequency. In addition to that, there's loss of feedback control in the HPA axis. A lot of my early research was on stress. And what we found is that one of the central mechanisms for controlling the stress response also becomes impaired in the brain. What do you think that is? The hippocampus. So it turns out the hippocampus has a really important role as a coordinator of the stress response. It actually sits right next to the hypothalamus, and it sends dendrites right into the HPA axis. And it turns out that the hippocampus has more cortisol receptors than any other area of the brain. And the hippocampus does two things. One, it consolidates memory, and in particular the importance of memories, which makes sense. When you're under stress and the lion is chasing you, you want to remember that, and you want to remember that that was a threat, that that was bad. Right? So the hippocampus turns on, and it starts pinging the HPA axis saying, okay, start making more cortisol, we're running away or climbing a tree or fighting, right? Because the cortisol itself is saturating the hippocampus. But interestingly, if this continues, if people have ongoing stress, if you stress the brain from inflammation, or if you stress it because of emotional issues or repeated low-grade traumas, the hippocampus is initially swollen, and then cortisol, being a catabolic hormone, starts to degrade the hippocampus. Because what does cortisol do, right? It breaks down tissue. It breaks down the immune system. It breaks down the cardiovascular system. That's what it does. Well, it turns out it'll fry your brain. And in particular, when that hippocampus starts to shrink, remember it has more cortisol receptors than any other area of the brain. So it's going to start changing first. And when that hippocampus starts to shrink, other brain centers say, oh gosh, prefrontal cortex, amygdala, locus ceruleus, all these other brain centers say, woo, okay, brain on fire, too much stress, hippocampus is shrinking, and it shuts the whole stress system down. So individuals go from a high cortisol state to a low cortisol state. This is how people get into hypocortisolism, low cortisol. I hate to break it to you. And this is just one of these things. But it turns out when people have been under lots of stress, the truth is they haven't wasted their adrenals. It's that they fried their brain. Okay? This is called the neurodegenerative model of hypocortisolism. It's 30 years of research. And we focus on this too. We actually have ways, and we'll get into this at some point, where in fact we structurally measure the size of the hippocampus in our patients. And I can show you very small hippocampi or swollen hippocampi in Lyme and mold patients. It's why their cortisol levels are so flat. The brain has said enough is enough. Now, interestingly, even though there's been this sort of label and nomenclature of adrenal fatigue and all this sort of stuff, where do you think ginseng works? In the brain. Where do you think rhodiola works? In the brain. Where do you think ashwagandha works? In the brain. Where do you think maca works? In the brain. Do you think ginseng works at the level of the adrenals? No. Very little activity. So you are treating the brain, right? When you're doing your sort of stress adrenal support. That's what you're actually treating. This is why adaptogens and tonic herbs were considered and elevated to be really special in most herbal traditions around the world, because they reduce neuroinflammation, they reduce neuroexcitation, and certain extracts in particular actually induce repair of the brain. So this is what we're dealing with, is a brain on fire, an immune system that's been triggered, and people can't turn it off. And then there are these secondary and tertiary consequences because as MSH drops, 12 different other things go wrong. Right? So this is why this is such a complicated multi-system, multi-symptom illness process. So 2008 definition. Yes, potential for exposure, symptoms similar to those in the published literature, labs similar to published literature, response to treatment. This is how we built our intellectual architecture. So this is our data. And so what about the deeper aspects? So in some sense, what I just began to describe for you would be a layer of blood labs, a layer of expression in the metabolism called the proteome. These are proteins that are being expressed as a result of an inflammatory insult. So these are our proteomic labs, C4A, MSH, so on and so forth. But we had a suspicion that something else was going on with patients as well. So we started doing a little bit of extra work. And we knew that the body follows a natural order. And we thought about this in terms of, well, what if things are happening out of sequence? What if there are signals that are going on at the level of the genome that is part of this inflammatory chaos that's creating abnormal genetic signaling? Well, what would that look like in nature? Well, we know that we're in the fall and the leaves are starting to turn and the geese are flying south, and we know that the winter's coming, so all is in order. But what if the geese leave too soon? Or what if the animals don't prepare? This would be an environmental disaster that if things don't happen in a certain sequence in a coordinated fashion, this is actually when disease arises. And so is this a reflection of that? And the answer is, at the local level inside the human with these biotoxic exposures, that's exactly what happens. There's an additional epigenetic influence on these patients, which triggers this whole cascade of abnormal gene response, and that we have to deal with that as well. So it turns out that clinically, our geese are flying south too soon in this instance. And we have to restore normal, natural cycles internally. And I'll talk about that in a little while. So this gene transcription is what we're really discussing. And that is DNA that makes RNA. And it's at that level where we're looking at how genes are functioning in this patient population that has really become abnormal. So when DNA makes RNA, that's called transcription. And it turns out that, this is initiated by these toxins And, mold is something that we can potentially live with, but even when it's threatened, it starts to create mycotoxins. That sometimes it's dormant and sometimes it's active. So all creatures obey these laws, including mold. And so if, nature depends on this normal order, and it does, what would we call abnormal gene transcription? Meaning, what happens if DNA is making RNA inappropriately? Does disease arise? And this is the new language where we're going from genetics to genomics. So this is the area of what we call molecular biology. Genetics is and was the effort to sequence the human DNA. And that was really identifying the structure of genes. What was there? That's what we first wanted to know. And I'm sure some of you have ordered 23andMe and some of these other, genetic sequencing, and that's what you're doing. You're basically just measuring the structure of the DNA. And you're asking yourself the question, is there heterozygosity or homozygosity? Is there some alteration in the gene sequence that might be somehow impairing an aspect of metabolism that in turn is leading to symptoms? So, for example, MTHFR. So let's slay that one too. We've already slayed adrenal fatigue, so we're going to go slay MTHFR as well. I hate to break it to you. So genetics to me is like the components of building a guitar. It's got a rosewood body, it's got steel strings, frets, a neck, and that's genetics. That's all it tells you is that, yep, I'm looking at a guitar. And the SNPs that you can get from various gene sequencing companies just tells you whether or not the components have changed. Is there a different kind of wood? Or maybe there are nylon strings, right? And maybe that is important in terms of the difference between the two. But what you don't know is what's the music that's being played off the guitar. What's the harmony? What's the melody? Which genes are on and which are off? That is genomics. And the only way you can know that is not just measuring DNA, but you have to measure RNA. And then RNA in turn makes proteins, so that's the proteome. And then the proteins then glom together and fold, and they make small molecules. So we have the genome, we have the transcriptome, which is the RNA. We have the proteome, C4a, MSH, others, so proteins, and then we have the metabolome, which is all the small molecules of the body. This is the new language of molecular biology. We've gone from genetics to genomics. We've gone from let's build a guitar to let's play some music. And it turns out that just knowing the structure of what's there doesn't really tell you a lot about the music. So we started looking at that as well. So does a genomic perspective change how we look at chronic illness? And we would say absolutely yes. That it's not enough to get patients normalized through blood labs, but that in fact when you have a tool of being able to measure the genome, the transcriptome, to see what music is being played, is it now full of harmony and melody or is it still disordered? What will happen with patients is that you can get them better to some degree, but then they go back. And the reason why they go back is underneath all of that improvement, the orchestra was not playing properly still. That we have to find ways to restore normal gene response to get that harmony back again. And if that doesn't happen, these patients remain chronically inflamed and chronically ill. So this host genomic response to initiation of injury becomes the disease. So what can we say about this multi-system, multi-symptom illness? Chronic fatigue, fibromyalgia, post-Lyme show harmony of normal gene transcription? No. We can see disorder at the genomic level. Has nature gone awry? Yes. And I'm going to walk you through all the pieces of this. So we're going to go from symptoms to proteomics to transcriptomics to actually end organ consequence and put all these pieces together. So what is altered in this activity of gene transcription? They have chronic inflammation. It's a response to injury. And it's tied to these abnormal transcription factors. So, it turns out microRNA are part of the signaling system for turning on and off gene expression, just as important as methylation. So there's all sorts of epigenetic influences that occur around the genome. MicroRNA being one, long non-coding RNA being another. Interestingly, we now have products that actually are rich in very positive transcription factors in their ability to sort of collectively modulate gene expression. And so this notion of regulating gene response is really critical. So, CIRS, Chronic Inflammatory Response Syndrome, abnormal proteomics, abnormal transcriptomics, abnormal regulation of immune function, abnormal regulation of hormone function, and these diseases are all around you. And in fact, they're in this room. I guarantee it. And on a personal note, I'm one of them. I'm in the 20%. I'm very mold sensitive. And so, I know what it's like to sort of live that experience of, having to first not understand, why are you getting sick when you'd been healthy for a long time? And then why all these other therapies don't really work until you walk through other doors. And so it's really common. But the good news is, the silver lining is, however complex this seems, it's treatable and fixable. And we've shown that too in our studies. So common, are these illnesses 21st century? I used to think so. Maybe we're just, it's an environment in which people are nutrient deficient and they're under more stress and they're exposed to more toxins, and maybe that's what this is. It's just the conditions of our state of life today. Well, it turns out that I was wrong, that this pathophysiology around lack of gene regulation that affects these ribosomal structures that are evolutionary old, meaning 3 billion years. This illness has been around a long, long time. And there might be sort of adaptive advantages that we haven't figured out why this occurs. And just to be clear, because I'm really big on definitions, DNA undergoes transcription, we make RNA. RNA is the mirror of DNA. That's the signaling molecule that is expressed off an active gene. RNA then goes to what's called a ribosome. This is an intracellular subunit that then absorbs the RNA and then translates it into a protein, right? So ribosomes are the engines of protein production, and they rely on RNA to do that. So back to a case definition. CIRS was certainly found in water-damaged buildings, so we know that when mold is growing, these patients can get really sick. But it's also found in other things, Ciguatera, cyanobacteria, algae such as Pfisteria, dental issues, all sorts of biotoxins. And sure, Lyme disease, but also recluse spider bites will initiate this chronic inflammatory response. Even reactions to some immunizations and vaccinations, anthrax being one of the classic, can initiate a CIRS reaction. We also see it in traumatic brain injury, that if you shake the brain, it can initiate this inflammatory response. And in the 20%, they can't turn it off. Part of my work looks at this issue of what are all of the factors that are leading to, in particular, this neuroinflammation, this brain on fire phenomenon. One of my co-researchers is one of the top traumatic brain injury specialists in the military. He's a Navy physician. We work with Navy SEALs. And what we have found is that, just like in any other population, 20% of those guys carry these genes. And that if they have repeated concussive events and they have exposures to infections, which of course they do, and then they're living in moldy environments, which they do, they can establish this chronic inflammatory response that can ultimately affect and damage their brain. You have to ask yourself, is there a potential for exposure? Well, musty smells are okay, but oftentimes patients are living in an environment where they don't smell anything. They say, oh no, I don't have any mold. My house was built last year. What we want to do is PCR. This is very precise testing on the molds that are in the environment. What we don't want to do are air samples. However much your local company wants to tell you how accurate air sampling is, it's terrible. PCR is not perfect, but it's pretty good. So we have patients who has actually swabbed their home for mold. We want to capture mold spores that have settled on dust. That's a more accurate way of assessing for mold in the environment. So what's the potential for exposure? Bacteria, soot, things that smell funny. Well, you don't want to guess. You want to use PCR. But the problem is so many things make people sick. And sometimes there's no specific causation, which is what makes this illness very frustrating. You can't really tell. Is it home? Is it work? Were they bit by a bug or a spider? What turned all this on in the first place? We don't have all the answers yet. We have some, but not all. And so a focus on any single entity is sort of wrong-headed. It makes it frustrating, but it's just the current reality. So at least 30 entities that make illness from inflammation in this category. I'm sure you've heard of all of them and you check for them in your practices, right? Beta-glucans, mannins, spirocyclic diamines, lipopolysaccharide, actinomycetes, hyphal fragments, cell wall fragments, bioaerosols, endotoxins, MVOCs, mycotoxins, and so on and so forth. It goes on and on and on. Unfortunately, there's enough stuff out there in terms of organisms that can trigger that innate immune response that sometimes we just don't know what did it. Sometimes we do. So, as CIRS, we have a wealth of biomarkers, which we've confirmed. We've published about 25 papers, over 4,000 patients, including children. Multi-site, so we have a number of clinics that collect data in this regard. We use ancillary data to confirm, and then we also add in other components. So we'll look at cardiopulmonary testing, VO2 max, elements of dysautonomia, so gastroparesis or gallbladder dysfunction, for example, or poor gastric motility. We also look at brain-related changes, and then, of course, all the blood labs, the proteomics, and in particular, the immune markers. And also really simple tests. It turns out that, interestingly, one of the consequences of a brain on fire is that we lose our contrast capacity. And you say, well, what is that? Well, it's actually a color discrimination. So people have a hard time distinguishing among shades of a similar color as their brain becomes more inflamed. And the way to test for that is something called a visual contrast study. I'll go over that tomorrow. VCS test was developed 50 years ago, really well reliable, takes 10 minutes, it's $15 to do. And if they have a brain on fire, they're likely losing contrast sensitivity. 90% of people who are in this category will fail the visual test. 10 minutes. You can know just like this. If they have lots of symptoms and they fail their visual test, you have a 98.3% sensitivity that they're a CIRS patient. That's it. That's all you need to do. Symptoms, visual test. 98.3%. Now, remember, 10% of people can pass the visual test, no matter how inflamed they are. So by passing the visual test, you can't necessarily confirm, but it's part of our diagnostic criteria. And so we look at all of this, right? In addition to that, we have treatment protocols that I'm going to go over in detail. I'm not going to talk at a 30,000-foot level. That's just today. We're going to get into all of the different steps of assessment and treatment. But we end with something very special, because you might say, well, Dr. Heyman, you talked sort of about genomics and all this fancy stuff and microRNA and transcriptomics and, okay, the genes have gone wild. What do? We actually have a therapy for that. But there's no difference between a water damage building CIRS patient and other types of patients, right? Because they can have dinoflagellate exposure, again, to Pfiesteria, which was an algae bloom in the Chesapeake, Ciguatera, which is an organism that lives in fish and shellfish, apica complex sands like Babesia, which of course comes from tick-borne illnesses, cyanobacteria. Sometimes we don't always know. But again, the symptom list is identical. One thing I would caution you against, as we've done our statistical analyses on patients and we've looked at them across populations and across exposures, the one thing we can say for certain is, based on symptoms alone, you cannot guess what your exposure is. And all of the symptom lists about, oh, well, that's a Babesia symptom. Well, no, you have joint pain, so that has to be Bartonella. Or you have brain fog, so that must be Lyme disease. Or wait a minute, you have stretch marks on your back and cherry angiomas. Well, that's Babesia. No, it doesn't work like that. The body has this very coordinated, elegant response to the exposure, which can generate a whole long list of symptoms and problems. But you can't tell based on how they feel alone what the exposure was. So let's do a little digression into Lyme disease just for a second. So not easy to deal with. And the first challenge is sort of, what is the landscape almost politically in terms of what do we accept as a case definition? And we know that in sort of the standard infectious disease community, they don't believe it goes chronic. They believe that there's a rash in over 70%, the erythema migrans rash. They rely on the two-tier testing, which they claim is sensitive. And they say there's no such thing as seronegative Lyme, meaning negative lab test, but they definitely have Lyme. They don't believe in that either. And then the use of antibiotics for over 4 weeks is not recommended. Okay, so that's the standard of care. That's what the AMA brochure is going to tell you. When your local friendly infectious disease MD doc comes into your office and says, this is how I want you to understand Lyme disease. Okay? Now, there's another group, a competitor, ILADS, the International Lyme and Associated Diseases Society. This is sort of what are self-described as the Lyme literate physicians. And they have a different perspective. When they've gone to the literature and how they've treated patients, they say chronic is common. The rash only exists in a minority of patients. They agree that there's poor sensitivity to the normal testing, so we miss a lot of Lyme patients. They also believe in seronegative, that you can be infected, but the test is negative. And that they also recommend, as appropriate, longer courses of antimicrobials and antibiotics. So you have these two extremes. You have one group that says, hey, Lyme is easy to diagnose and treat, and it's only 4 weeks of antibiotics. And then this other extreme that says, oh my God, it's everywhere and everybody has it, and I know your labs are negative, but you need to be on antibiotics for the next 3 years or longer. Right? So it's somewhere, this is all you get in Lyme. The sky is falling or no big deal. Right? That's it. And, our perspective is they're both right and they're both wrong. In probably 80% of the population who doesn't carry the HLA sequences, 4 weeks of an antibiotic is probably good for most of them, but not all. Interestingly, when you look at the Lyme literature, everybody agrees on the one thing that says there is something called post-Lyme syndrome, that in 20% of the population, they'll have persistent symptoms forever. But then they go on to say, well, we don't know why. And we keep saying, our group keeps saying, well, we know why. It's the HLA, and it's the chronic inflammation. We have an answer. And it's very persistent in the literature, 20% chronic Lyme, post-Lyme syndrome. So I also agree that the other extreme can be right too, that there are clearly some patients where they have persistent symptoms, and they need a longer course of therapy and strategy to sort of clear the antimicrobial deck. That's fine. But the one thing you better be doing at the same time, which most of them don't, is turning off the inflammation, correcting the end organ damage, returning their genomics back to normal. Because if they don't do that, they're going to be on the antimicrobial wheel forever. Who's seen this in their practice? Who's seen patients where they just got stuck with the label of Lyme disease and they're on it forever? How do we turn it off? Well, I'm going to give you a model for how to do that, a language, an approach. I'm not saying it's the end-all, be-all, cure-all for everything, but we've solved some pretty sticky issues in that regard. So yes, they agree that there's this thing called chronic Lyme disease in the ILADS population, that these are patients that require long-term therapy, and that there's different categories of that, late untreated or post-treatment that failed. Essentially, so either they've had it and no one knew, or they were treated at some point and they still had persistent symptoms. Okay, so what are some of the tools that are now available for better diagnosis of Lyme? So if you haven't heard of this before, there's a new antigen test that's on the market, and it's a nanotrap test. So basically, it uses nanospheres to capture an antigen that's expressed on the outer coating of Borrelia. Turns out that all pathogenic forms of Borrelia excrete something called outer surface protein A, and it finds its way into the urinary tract and out into the urine. So this is a direct test. Think of it as now a test that's actually literally looking for bits and pieces of bug in the urine, as opposed to an indirect serology test, which is called a Western blot. Now, I hate to break it to you, but a lot of the other tests that are on the market now are here on serology, which are, they have issues. And there's a lot of popular ones that patients like to use where they get told they have Lyme disease, but it turns out that the sensitivity and the assaying are probably a little indiscriminate. So the one thing I know is that if you have outer surface protein A in your urine, you've got Lyme. We found a bitten piece of a bug. There's no denying. Now, is this test perfect? The people who developed the test would like to say it's excellent that it has high sensitivity and certainly absolute specificity, meaning that not only will they detect it 100% of the time, but they're 100% certain that it's Lyme disease. And they've used very sophisticated verification models. They took 24 patients who had known Lyme, rash, the whole thing, and all 24 tested positive on this nanotrap. Okay, so that's pretty good, 100%. But we know that it's not perfect. It's better than what's out there, but there's a big asterisk, and that is, what about patients that have been treated? Let's say the Lyme is in its cyst form, it's kind of curled up, it's not expressing a protein. We don't know all the test characteristics. But I can tell you in an untreated population that the Lyme is happily making protein and shedding that outer surface protein A, this test is pretty good. Best one I found on the market. So let's go back to CIRS. Symptom cluster analysis, really key. We also do pulmonary function testing with diffusion capacity because, of course, they get interstitial lung disease as well as restrictive lung disease. We also see loss of VO2 max. So if you have individuals that were highly trained athletes and now they can't run from here to there without falling down and passing out from exhaustion, you'll see these patients developing low exercise tolerance, poor pulmonary or poor peripheral blood flow. They'll fail their visual test. NeuroQuant, which I'll talk about tomorrow, is a brain scan that we do where we show architectural changes in the brain. Then we run our transcriptomics so we can actually look at the music of the genes. And then we have confirmed our interventions that we show at every step of therapy, all of these markers going back to normal in a sequence. So what are these symptoms? So not every, we really want you to sit down and actually go through the symptom checklist. You guys are really skilled, and we need you to sort of make sure that you ask all the appropriate questions in this patient population. So what are the symptoms? They are actually grouped into 13 categories, and we found that if patients answer yes in at least 8 of these 13, they begin to meet criteria. And some of the more common ones, fatigue, headaches, aches and cramps, electric-like sensations, light sensitivity, blurring, tearing, shortness of breath, abdominal pain, secretory diarrhea, joint pain, AM stiffness, a lot of cognitive issues, a lot of word-finding difficulties, short-term memory loss, obsessions, compulsions, depression, anxiety, mood disorders in general, sleeplessness, all sorts of neurologic issues related to a brain on fire. Mood changes, loss of appetite control. Interestingly, as the brain becomes more inflamed, there's damage to leptin receptors, which control hunger and weight control. These patients can rapidly gain 30, 40, 50, 60 pounds that is unresponsive to lifestyle interventions. And it's because the leptin receptors are becoming damaged. Leptin can't dock in. They can't control their appetite or their weight. And so if you have an individual that's really frustrated, they've been, oh gosh, paleo, vegan, ketogenic, right, for the last 4 months, and they're doing CrossFit every day, as well as training for a triathlon, and they're not losing any weight, you think CIRS. You think CIRS. If they're urinating all the time, I mentioned this, numbness and tingling, a lot of peripheral neuropathies, and then vertigo, tremors, and skin-related issues. These patients get weird muscle vibrations, cramping, twitching, creepy crawly sensations, burning, a lot of balance issues and instability, running into walls, all sorts of things. Okay? So the cognitive and neurologic issues are huge, and it's from the systemic inflammatory response. What's happening is the blood-brain barrier is being bridged by these inflammatory proteins, so there's degradation of these very beautiful linings in the body. So the gut lining, the endothelial lining, and the vascular system, and then the blood-brain barrier lining as well, single cell layer thick. We see this innate immune activation, which I've mentioned, absence of these regulatory neuropeptides from the brain, disorder in different aspects of the immune system, which I'll cover tomorrow, and that all of this has been well documented. So we've confirmed this by 2 double-blind placebo-controlled trials, and we've published, and it's been confirmed by other authors as well. And before our transcriptomic work, we were still kind of feeling our way through the system. We want to set a baseline set of labs. There's a protocol for each step in the sequence. I don't want you to skip steps. And it's because I don't want you to paint a house that's already engulfed in flames. So if it's a body on fire and lots of inflammation, you can't do the special final step of therapy, which is the VIP spray. It won't work. You can't get reorder of gene activity if the body is still sick. So there's an order that we go through. Labs won't ensure cure, meaning, remember, symptoms have gone away, blood labs have gone away, but we still found abnormalities at the genomic level. So that's another key, that there's a process to this, and that the labs alone, normalized labs, won't ensure cure. There's even deeper work to be done. Okay, there's organ damage, and there's genomic aberrations. So symptoms point the way, but we knew that there was a lot of cognitive issues, so we started imaging patients' brains. And we used a novel technology that allowed us to measure areas in the brain by volume or size called NeuroQuant. And this is a volumetric brain scan that precisely through mathematical modeling allows us to take a look at 11 key centers. And it turned out that there were signature patterns of injury that had not been identified in any other area of neurology, that the brain changed in very unique ways due to these biotoxic exposures, that we found a fingerprint of damage to the brain that we then can use to help determine the original exposure, but also to show cure, because we want the brain scans to go back to normal as well. So again, all that cognitive decline because of the memory loss, concentration issues, word finding difficulties, confusion, disorientation, again, not specific for a biotoxin. You just don't know. But we did find the fingerprint, and we showed that VIP not only corrects the genomic issues, it also corrects the brain damage. So just by sealing up barriers doesn't work alone. You're still left with damage to the organ tissue. And so yes, I have my patients, they make their chia seeds and ground flax and their almond milk in the morning because that's very nourishing for all the linings in the body to kind of seal everything up. But that alone, that sort of approach, is not going to get you all the way with these individuals. So we know that this brain fog that a lot of patients talk about is from this chronic inflammation that they have. And it's because there are these changes at the architectural level. There's dendritic connections called pruning, where the dendrites start to retract in and of themselves. And they're withdrawing away from the inflammatory insult. So we see volume loss in patients. Actually, the brain shrinks in key areas. The problem is we don't know if it's pruning or atrophy. Pruning is volume loss, where if you take the insult away, dendrites regrow. Atrophy is dendrites don't regrow. It's true brain damage. And we won't know until we treat patients whether or not that we can get regrowth. And I have some other tricks that we use along the way too that I'll talk about to protect the brain. So this is a recent innovation. Basically in the 1990s, total brain volume was being measured, but then in 2000, we can get really detailed with specific areas of the brain. So what do I need to know to treat? There's no single therapy which corrects the blood-brain barrier pruning. Multiple sequential steps based on this biotoxin pathway, which we'll go over. A relapse with re-exposure wreaks havoc with sequential intervention, meaning if you're getting them to a certain point and then they go and travel to a hotel and they experience some mold, they go all the way back. And now the body's excited again, and we have to deal with that. We also use our next generation sequencing. We're looking at 25,000 coding genes, 25,000 non-coding genes. We thought some of it was just DNA junk. It's not. Long non-coding and microRNA are really important to this story because they regulate how genes function. So we're looking at that role of microRNA and long non-coding RNA. And what we're really evaluating for is regulation of gene expression. And again, we have ways of altering that. And here is the nomenclature soup. So if you were really worried about, oh my gosh, CBS, COMT, MTHFR, MTRR, right? I think some of you are sort of exposed to just how we build a guitar. MAP2K, Chaperone, GRP78, CYP17, CYP1A, CYP, NR50A1, NRB1A. Note the conversion of active T2 to inactive 3-hydroxy H22. Gliotoxin block NF-kappa beta, lowers TNF alpha, increase COL1, OCN, BSP, RUNX2. You get the gist, right? You know all this? We're going to go over this tonight after dinner. Just kidding. So these are all of the proteins that are expressed with gene response. And as we map these pathways, we can collect thousands of data points on a patient about which genes are on or which are off, and we can see all the way out to the final metabolome what's occurring. So we've applied transcriptomics to this patient population. But there are treatment challenges. So we need to eliminate the exposure. First, you have to decide if it's still there, because of course sometimes they had Lyme, they got treated, and they're just still sick. Don't get confused with symptoms. You have to correct this inflammatory response. You have to repair the nervous system, and you have to restore normal genomic activity. All of it. 20% of the population. Headaches, fatigue, brain fog, cognitive decline, mood disorders, joint pain, digestive complaints, dysautonomias, cardiovascular issues, obesity. You guys don't see any of that in your practice, do you? So, this is where we're going. I always like to kind of give a preamble, which is to say we're going to go through all of this. This is our biotoxin evaluation process. And yes, I'm incredibly rudimentary with my slide development. I wish I was very, artistic, but instead I can just do boxes and stuff. So we're going to go through tomorrow the initial screening. We're then going to verify these patients in terms of all the tiers of testing, and then diagnostic refinement to say, well, what is the exposure and is it still present? This is the coordinated, reduced set of therapies that are directed at the biotoxin patient. This is very non-integrative. It's not very amenable to our community. It's mostly for physicians to correct these abnormalities. This is called the Shoemaker Protocol. It's been well published, and it's a stepwise set of therapies to correct all of this disorder that's been accruing. But this is what we're going to go over. This is an expanded, more integrative model for dealing with CIRS. So once you make the diagnosis, this is our treatment path. We're going to go over foundations, and then exposure elimination, reduce the toxin burden, deal with their metabolic balance, and resolve and repair. This is how I conceive of that triangle in a language that we understand that's still evidence-based. So every treatment step has a purpose to get you to where you want to go. And I'm going to make one big apology. There might be some people in the room that can't prescribe medications. And some steps along the way require a prescription. I have not figured out how to get to the end completely without doing this totally naturally. So I want to just apologize up front. We've worked really hard to get it to where it is. And you might just need to, if you can't prescribe, you might need to collaborate with a local provider who's amenable enough to help you down this process. And there's tons of research now. We can send you research papers and they can educate themselves. And if you want, I'll get on the phone and talk to them and say, prescribe the VIP, fix their brain, fix their genetics. So sometimes we just, there's only so far we can get. But I want you to understand, we've worked really hard all summer long to figure out what would be the easiest path forward for as many practitioners as possible. So we've gone from this, which was not very integrative friendly, to this, which is sort of integrative medicine 2.0. Okay? But we're going to get you there. I promise. So, what did you hear today? A definition of CIRS, which is this chronic inflammatory response. 20% of people have it because of their genetics, and they can't turn it off. And when they can't turn it off, it fries their brain, and it means that their genes are activated, and we have to do something about that too. And I talked about symptom clusters and also something called proteomics, which are just the proteins in the blood. But if all you remember for today is that we're dipping our toe in the water of a big group of patients that you see every day in your practices that are really, really sick, and that it's because of chronic inflammation, that's all I want you to understand. We're going to go through all the details tomorrow. So we're going to go, the next talks are this, and this. Okay? So all the details. Okay? Easy. I hope you get some rest tonight and eat really healthily because this is going to be a whole new world for you. Okay? And as I said, when you walk through this door, you're not going to go back. All right? So thanks a lot for your attention, and I'll see you tomorrow.